So welcome you all to this lecture. So today's topic is identification of bacteria. So once you receive a specimen in the microbiology laboratory, you will be subculturing the specimen. You will be culturing the specimen onto certain culture media, and you will get a colony on the culture media if there is any infection. So based on the colony morphology and certain biochemical tests, we can identify the bacteria from the culture. So that is how we used to do conventionally using certain set of biochemical tests. And nowadays, there are automated techniques available in which the machines will give the identity of the bacteria. It is a quicker method. So recently, we are using automated methods also. So first, coming to the routine conventional biochemical identification. So it is based on the type of the colony morphology, the gram staining appearance, etc. We will decide on the biochemical test to be employed. So first we will see the morphology of the colony, and based on that we will get an idea what type of organism it is. Then we will do a gram staining, and based on that you will get an idea whether it is a gram positive or a gram negative organism, whether it is a cocci or a bacilli. Like that, based on that we can decide on the appropriate biochemical test. Initially, the two tests that we do are catalase and oxidase test. These are the first tests that we usually do after doing a gram staining. Then for gram-negative bacilli, additionally, we will be doing a set of biochemical tests, which include indole, triple sugar ion agar test, then urea hydrolysis test, citrate utilization test, and nitrate reduction test. These are certain biochemical tests which we will do commonly for gram-negative bacilli. Then for gram-positive cocci, there are certain other tests which are known as coagulase test for Staphylococcus aureus, then bile esculin test and heat tolerance test. It is usually done to identify Enterococcus. Then inulin fermentation test done for identification of pneumococcus. Also for pneumococcus, we can use bile solubility test. Then for group B streptococcus, we can use another test which is known as Christie-Atkins-Munch-Peterson. That is CAMP test. That is a test which is used for identifying group B streptococcus. So after identification of the bacteria, you have to do the antimicrobial susceptibility testing. Using the antibiotic disc also, we can identify the bacteria. So after this biochemical test, anyway, we will be putting antimicrobial susceptibility testing. So while putting the antibiotic disc, we can use certain special disc which can be used as identification disc also. So examples of such disc are optotion susceptibility test. It is done for differentiating pneumococcus from streptococcus viridans. So pneumococcus is sensitive to optotion, while streptococcus viridans is not sensitive to optotion. Then bacitracin susceptibility test, it is used to differentiate group A streptococci from group B streptococci. Both will be hemolytic, and group A can be differentiated from group B by bacitracin test. Group A will be sensitive to bacitracin. Group B will be resistant to bacitracin. So these are certain antibiotic discs that we can use for identification of bacteria. So we are going to individual biochemical test. First one is catalase test. So catalase test, it is done to identify the presence of catalase enzyme in the bacteria. If the catalase enzyme is present in the bacteria, the test will be positive. So to perform this test, we use 3% hydrogen peroxide. You can do in several methods. The most common one is the slide catalase test. So we'll take a slide, and in that we will add a drop of hydrogen peroxide. 3% hydrogen peroxide is added. Then we will take the colony of the organism, and it is added onto this 3% hydrogen peroxide. If the organism can produce catalase enzyme, it will break down hydrogen peroxide to liberate nascent oxygen. This nascent oxygen will produce bubbles. So the catalase test positive will appear as bubble formation or effervescence. So this one is negative, no effervescence. So this test, it is used to differentiate between Staphylococcus and Streptococcus. Staphylococcus will be showing positive catalase test and Streptococcus will be showing negative catalase test. Mainly it is used to differentiate Staphylococcus and Streptococcus. And there are other catalase positive organisms also. Examples are Enterobacteriaceae, Vibrio, Pseudomonas. All these are catalase positive organisms. The second one is oxidase test. So in this test, we are detecting the presence of the enzyme cytochrome oxidase present in the bacteria. If this cytochrome oxidase enzyme is present in the bacteria, it will catalyze the oxidation of reduced cytochrome by atmospheric oxygen. So this enzyme, the main function is it will catalyze the oxidation of reduced cytochrome by atmospheric oxygen. And what we are using is called a oxidase reagent. This oxidase reagent, it is chemically tetramethyl paraphenylene diamine dihydrochloride. So it is tetramethyl paraphenylene diamine dihydrochloride. So we can do it in a filter paper strip. So nowadays commercially the oxidase reagent is impregnated onto the filter paper and it is dried and it is available in bottles. Otherwise, you can take a filter paper and you can use a freshly prepared oxidase reagent. It will be available in powder form. You can prepare it by diluting with water and you can use this freshly prepared oxidase reagent. Add a drop of this reagent onto the filter paper strip and take the colony of the organism using cover slip or a wooden stick or anything. You don't have to, you shouldn't use the loop or the straight wire for performing the oxidase test, also for catalase test. So you have to use either a cover slip or a glass rod or a wooden stick you can use. Take the colony using this wooden stick and rub it over the filter paper having the oxidase reagent. So if the organism is oxidase positive, it will give a purple color immediately after rubbing this colony on the filter paper containing the oxidase reagent. So it should appear within 10 seconds. So a deep purple color will appear in 10 seconds if the organism is oxidase positive. That is because of the conversion of tetramethyl paraphenylene diamine dihydrochloride to a substance known as indophenol blue. And if it is negative, it will be appearing like a yellowish color or no color change is seen. So oxidase positive examples are Pseudomonas, Vibrio, Neisseria, Bacillus, etc. All these are oxidase positive organisms. And oxidase negative organism are members of family Enterobacteriaceae, Acinetobacter. All these are oxidase negative organisms. The third one is indole test. So this test, it detects the ability of bacteria to produce an enzyme known as tryptophanase. So if the enzyme tryptophanase is present in bacteria, it will break down the amino acid tryptophan present in the medium into indole. So we are using peptone water to perform the indole test. So the peptone water will always be having the amino acid tryptophan in it. So if the bacteria can produce tryptophanase enzyme, it will break down the tryptophan present in the medium to produce indole. So the reagent that we are using to do the indole test is Kovacs reagent. So if the bacteria has converted tryptophan into indole, this Kovacs reagent will combine with the indole to produce a cherry red color. So that is how the reaction works. So Kovacs reagent, it combines with the indole to produce a cherry red color. This red color will be formed near the surface of the broth. So it will be seen as a ring on the surface of the broth. That is how the indole positive reaction will look like. Examples are Escherichia coli, Proteus vulgaris, and Vibrio cholerae. All these are examples for indole positive organisms. So this is how the indole positive reaction will look like. A red colored ring is seen at the surface of the broth. Negative means it will be appearing like a straw colored appearance only. It's not a red colored ring. So indole negative, a yellow colored ring or a straw colored ring is formed near the surface of the broth. Examples are Klebsiella pneumoniae, Proteus mirabilis, Pseudomonas, Salmonella. All these are examples for indole negative organisms. Now the next one is citrate utilization test. So citrate utilization test, it detects the ability of a bacteria to utilize citrate as a sole source of carbon for their growth. So if the organism is utilizing only citrate as a sole source of carbon for the growth, what will happen is then as the end product, some alkaline products will be produced as the byproduct, end product. So there will be alkaline metabolic products produced, and this alkaline nature of the media, it will convert the color of the indicator from green color to blue. So in this citrate utilization test, we are using an indicator called bromothymol blue. Bromothymol blue is the indicator used for citrate. So you are performing it in a media known as cement citrate medium. So if the organism has utilized citrate as a sole source of carbon, the alkaline byproducts which are produced, it will convert the color of the indicator, bromothymol blue, from the original green color to blue color. So the color of the positive reaction is blue color. If it is negative, it will remain in the same green color as that of the medium. So this is the citrate positive reaction. It is blue color. And if it is citrate negative, it is green in color. Examples for positive citrate reaction, it is shown by Klebsiella pneumoniae, Citrobacter species, Enterobacter species, etc. So they will show citrate positivity. Negative for Escherichia coli, Shigella, etc. All these are examples for negative citrate reaction. Then the next test is urea hydrolysis test. So in this test, we are testing the ability of the bacteria to split urea to produce ammonia. So to split urea to ammonia, the organism should be having an enzyme called as urease. So if urease enzyme is present, the organism can split the urea present in the medium to produce ammonia. So again, ammonia is alkaline. So the color of the indicator will change from the original yellow color to a pink color. So the change of color is from yellow to pink color. And the indicator that we are using is phenol red indicator. Phenol red indicator, it will change the color into pink color in alkaline medium. And the media that we are using is Christensen's urea medium. It contains urea. So this is how the urease positive reaction will look like. It is pink in color. And if it is negative, it is yellow in color. And the urease test is positive for Klebsiella pneumoniae, Proteus species, Helicobacter pylori, Brucella, etc. And the urease test is negative for Escherichia coli, Shigella, Salmonella, etc. So urease test is positive for Klebsiella pneumonia, and most important is Helicobacter pylori and Proteus species. So they will be having urease test positive. Then urease test is negative for E. coli, Shigella, and Salmonella. Then the next test is triple sugar iron agar test. So this test is done in a medium which is known as triple sugar iron agar medium. So this medium is also known as a composite medium because in this medium we can test the various properties of the bacteria. So this media, it contains three sugars: glucose, sucrose, and lactose. These are the three sugars: glucose, sucrose, and lactose in the ratio of 1:10:10. So glucose will be less, sucrose and lactose 10 parts more. So the uninoculated TSA medium will be red in color, and this medium is poured as a slant and a butt. So when you are pouring the triple sugar iron agar medium, we will be making it as a butt as well as a slant. So it has got a butt portion. Butt portion is the lower portion in the test tube, and a slant portion will be there. And phenol red is the indicator that you are using in triple sugar iron agar. There is one more indicator that we are using, that is ferric salts. These ferric salts are used as an indicator for hydrogen sulfide production. So we can identify whether the bacteria is able to produce hydrogen sulfide. That is one property that we can make out from this media. And the main property that we are making out from this media is whether the organism is able to ferment glucose, sucrose, and lactose with production of acid with or without gas. That is the property that we are testing in this media. So after inoculating the media, we'll be incubating it at 37 degrees Celsius for 18 to 24 hours. So interpretation, it detects three properties of the bacteria, that is fermentation of sugar to produce acid, fermentation of sugar to produce gas, production of hydrogen sulfide. So these are the various reactions you can expect in a triple sugar iron agar media. So this first one, it shows the uninoculated media. This will be the color of the uninoculated media, and it is poured as a butt as well as a slant. So first one is A by A gas plus H2S minus. So A means acidic reaction. Why A by A it is written? It is because A in the slant, also A in the butt. That is how it is interpreted. So A in the slant by A in the butt. So acidic reaction in the slant, acidic reaction in the butt also. So the next one is K by A. K by A means K is alkali, A is acid. So this indicates there is alkaline reaction in the slant, there is acidic reaction in the butt. Again, we'll write gas plus or gas minus, H2S plus or H2S minus. This is how you have to interpret the triple sugar iron agar reaction. So you have to write what is the reaction in the slant by what is the reaction in the butt area, gas plus or minus, whether it is plus or minus, and H2S whether it is plus or minus. This is how you have to interpret the triple sugar iron agar reaction. So we'll see individually what are the reactions. So we'll start from here. K by K, no gas, no H2S. So K by K means it is fully alkaline reaction, which means there is no fermentation of sugars in this organism, sugars by this organism. So the organism that you have inoculated in this tube, it is not able to ferment either glucose, lactose, or sucrose. That is why the whole media is appearing as alkaline. So K by K, no gas, no H2S. So to interpret gas, you have to see whether there is any cracks in the media or where there is any lifting up of the media. You can see in the second test tube, there is lifting up of the media as well as there is cracking of the media inside. So this indicates the production of gas. So here there is no gas production. Hydrogen sulfide is hydrogen sulfide production is indicated by the presence of blackening of the media. The blackening of the media indicates the production of hydrogen sulfide. So here no hydrogen sulfide is produced in this last tube. So that is how K by K gas minus H2S minus. Then K by A gas minus H2S plus. That is K in the slant area, A in the butt area. Here you can see there is no color is seen in the butt area. But still you will be assuming it as an acidic reaction. And one more thing in between, if the media is alkaline, it will be in the pink color because it is the phenol red is the indicator that we are using. Okay, so since it is phenol red indicator, the alkaline reaction will be appearing as a pink color. That is why we are writing it as K by K when it is pink. And the phenol red will be yellowish in color in acidic media. That is why the color is yellow. Acidic reaction means the color is yellow. So in the second last tube, K by A is written. So in the slant area, okay, it is agreed. It is alkaline reaction only, pink color. And in the butt area, since there is production of hydrogen sulfide, we will be assuming that the area is acidic only. So in acidic media only, hydrogen sulfide production is also expressed. That is how we assume. So we are writing it as K by A. Then hydrogen sulfide production is there, gas is not there. That is how K by A gas minus H2S plus. Then the third one. Here you can see again Slant is alkaline K, but is acidic A. So K by A, gas is not there. But you can see a speck of hydrogen sulfide. Speck of hydrogen sulfide means little bit of hydrogen sulfide. It is there at the junction between the slant and the butt. That is known as speck of hydrogen sulfide. That is this tube reaction is interpreted as K by A, gas minus and H2S plus. Coming to the third tube, here again K by A, then gas minus H2S minus. No H2S production is there. Then this tube, it is A by A, gas is there, H2S production is not there, H2S minus. That is how you will be interpreting the triple sugar iron agar test. Now, why these different reactions are there for triple sugar iron agar test? That is because some organisms it can ferment glucose alone. Some organisms will not ferment any sugars at all. Some organisms will ferment glucose. Along with that, it can ferment lactose or sucrose. That is how various reactions are there for. That is why different reactions are there for triple sugar iron agar test. So I told you in the last tube, there is no fermentation of sugars. This organism is not able to ferment any sugars, not even glucose. The K by A reaction means the organism is able to ferment glucose, but it is not able to ferment lactose or sucrose. The organism is able to ferment glucose, but it is not able to ferment lactose or sucrose. That is why the butt is showing a acidic reaction, but the slant is showing a alkaline reaction. So in the initial part, because of the glucose fermentation, some amount of acid will be produced. So in the initial few hours, the slant may appear as yellowish, fully yellowish it can appear in the initial few hours. But we are incubating it for overnight. That is around 16 to 18 hours we are incubating. So during that time, what will happen? In the initial hours, even though it is fully acidic, later on what will happen because of the less amount of acid that is produced, the amino acids which are present on the slant area, it will undergo oxidative deamination. It will produce this alkaline reaction. Alkaline reaction on the slant. So this alkaline reaction, it will happen on the later hours of incubation. That is because the amino acids which are produced in the slant can undergo oxidative deamination, producing an alkaline response. That is how K by A is happening. So one thing you have to remember is K by A means the organism is able to ferment only glucose. It is not able to ferment lactose and sucrose. Then A by A means the organism is able to ferment glucose. Along with that, it is able to ferment lactose and sucrose or lactose or sucrose, either lactose or sucrose or both together. So that is why large amount of acids are produced. So even after the oxidative deamination happening on the slant is not able to become alkaline because of the excessive production of acid. That is how the reactions happen in the triple sugar iron agar test. So I told you the ferric salts are the indicator for production of hydrogen sulfide. So it will combine with hydrogen sulfide to produce certain black colored complex. That is why the hydrogen sulfide production is appearing as blackening of the medium. Then now coming to the examples of each reaction. So acidic slant by acidic butt, A by A. So more than or equal to 2 sugars fermented. Glucose, then lactose and sucrose or lactose over sucrose. Okay. So examples are A by A gas produced no H2S. Example is Escherichia coli, Klebsiella pneumoniae. Then K by A no gas, no H2S. Example is Shigella. K by A no gas, H2S produced in small amounts. That is speck of H2S, Salmonella type P. K by A no gas, H2S produced abundant. Example is Proteus vulgaris. K by A gas produced, H2S also produced abundantly. Example is Salmonella paratype B. K by A gas produced, no hydrogen sulfide production. Example is Salmonella paratype A. Then alkaline slant by alkaline butt, that is K by K. No gas, no H2S. Example is Pseudomonas and Acinetobacter. These are non-fermenters. These are non-fermenters. So that finishes triple sugar iron agar test. Now coming to coagulase test. So coagulase test, this test can be done in two methods, either a slide coagulase method or a tube coagulase method. So if you are doing it in a slide method, you are detecting the clumping factor or the bound coagulase present in the organism. And if you are doing in a tube method, you are detecting the free coagulase enzyme that is secreted by the bacteria. So that is these are the two methods for doing the coagulase test. So free coagulase enzyme, it can combine with the plasma protein coagulase reacting factor, which will convert fibrinogen to fibrin. So if the enzyme free coagulase is produced by the organism, it can combine with the plasma protein, the coagulase reacting factor, and it converts fibrinogen to fibrin. This fibrin will be appearing as a clot or a coagulum in the tube. So clumping factor, it is a bound coagulase, that is, it is present on the cell surface. It is a fibrinogen by the cell surface adhesion. So to do the slide coagulase test, what we will do is, we'll take plasma and we'll mix the colony onto the plasma. Or you can first make a saline suspension of the colony and add plasma. So what you will observe is the clumping or the clumping of the or the aggregates which are formed in the positive test. That is how the slide coagulase positive reaction will be. Clumps will be formed. Okay. So otherwise, if it is negative, there will be a milky suspension. Then tube coagulase test, how you will do is, you have to dilute the plasma. So the normal method that we commonly use is, we have to dilute the plasma in a ratio of 1:6 with normal saline. So 1 ml of plasma plus 5 ml of normal saline you have to make. And you have to take 1 ml of this and add on to a tube. You take 1 ml of this diluted plasma onto the tube and inoculate 2 to 3 colonies of the organism. 2 to 3 colonies of the organism onto the diluted plasma, and you have to incubate it at 37 degrees Celsius. Observe it after 1 hour, at the end of 2 hours, at the end of 4 hours. If there is no coagulum formed till 4 hours, you have to keep it overnight, incubated, and next day morning you have to see for the coagulum formation. So how will you observe for the coagulum? You have to just tilt the test tube and see for the clot or the coagulum that is formed on the tube. Even if you tilt it, it will not be falling like a fluid. There will be a clot which is formed. If it is negative, means it will be just a solution. Only the suspension will be there. There will be no coagulum formation. So this test is positive for Staphylococcus aureus and negative for coagulase negative staphylococci. Now coming to the sugar fermentation test. So we can identify the organism based on what all sugars it can ferment. So it detects the ability of the organism to ferment a specific carbohydrate which is present in the medium, producing acid with or without gas production. So what are the common sugars that are used for sugar fermentation test? They are glucose, lactose, sucrose, and mannitol. So glucose, lactose, sucrose, and mannitol are the common sugars that we use for testing sugar fermentation. So we will take these sugars, carbohydrates in the media. So the concentration of these sugars is 0.5%. And we will use an indicator also. Usually we will use antrax indicator. So the antrax indicator, it will be pink color in acidic medium. Or you can use a phenol red indicator also. It is yellowish in acidic medium. You can either use a phenol red indicator or an antrax indicator. So when the organism ferments the sugar, it will produce acid. So accordingly, the color will be changing. If it is antrax indicator that you are using, it will produce a pink color. If it is phenol red indicator that you are using, it will produce a yellow color. So in the media, you have to inoculate the organism and keep it overnight incubated at 37 degrees Celsius. Next day, you have to check for the carbohydrate fermentation. And one more thing that you can identify is whether gas is produced or not. So gas production is detected by using an inverted Durham's tube. So you can see here, these are the test tubes in which we are taking the media, and you can see one tube is put inside the main tube. That is, one small tube is put inside the last tube, and it is in the inverted position. So here again you can see the media has raised only till this area. Here there is gas production. So gas production is indicated by the presence of the gas production. It is detected by using a inverted Durham's tube. Okay, so this is the gas production. Let us see. In these tubes, there is no gas production. So there is color change to yellow. So this is a carbohydrate fermentation. It is showing the fermentation of the carbohydrate. This is the positive fermentation reaction. This is negative. Now coming to decarboxylase test. So this test, it detects the presence of substrate-specific decarboxylase enzyme present in the bacteria. That is, several decarboxylase enzymes are there. So various amino acids can be decarboxylated to the final products, to the alkaline byproducts. So we will add different amino acids in various media, and we will check the ability of the organism to decarboxylate the different substrates. So if this enzyme decarboxylase is there, it will break down amino acid to produce alkaline byproducts. So we are using bromocresol purple as the indicator. It will convert the change in color is when it is alkaline, it will change to purple color. So there is alkaline end products which are formed. The bromocresol purple will change to purple color. So if you are using lysine amino acid, it will be decarboxylated to cadaverine. Ornithine, the end product is putrescine. Arginine, product is citrulline. Now coming to methyl red test, there are two tests that we are doing in glucose phosphate broth. That is a media used for methyl red test as well as Voges-Proskauer test. These two tests we do in glucose phosphate broth. We use the glucose phosphate broth. In that you will inoculate the bacteria, and after 48 hours of incubation, we will add the methyl red indicator. When we are adding the methyl red indicator, if the organism is able to ferment glucose to produce stronger acids such as lactic acid, acetic acid, formic acid, etc., the pH will be maintained below 4.4. In such a case, the methyl red indicator will change from yellow color to red. So the methyl red indicator, it will change from yellow color to red. So the positive reaction is like a red color and negative will be yellow color. So the color changes because the organism is able to ferment glucose to produce stronger acids, and the stronger acids is maintaining the pH below 4.4. That is why the color changes there. When you are adding methyl red indicator, the whole media will be changing to a red color. Examples for positive organisms: E. coli, negative is Klebsiella pneumoniae. Voges-Proskauer test also we do in a glucose phosphate broth. We inoculate the organism in the glucose phosphate broth, and we will incubate it for 48 hours. And at the end of 48 hours, we will be adding VP reagent 1 and VP reagent 2. VP is Voges-Proskauer. Reagent 1, it is 40% KOH. Reagent 2 is alpha naphthol. So the principle of this reaction is a certain bacteria, it will produce acetoin as the chief end product of glucose fermentation. Once they ferment glucose, acetoin is produced as the chief end product. In the presence of alkali, so alkali is provided by VP reagent 1, that is 40% KOH. So in the presence of alkali and atmospheric oxygen, this acetoin, which is produced as a result of glucose fermentation, it is oxidized to diacetyl. And this diacetyl, it will react with the VP reagent 2, that is alpha naphthol, to give a red color. That is how the color is formed in VP reagent. That is VP reagent, VP test positive is red in color, and negative is again yellow in color. So the principle is certain bacteria can ferment glucose and produce acetoin as an end product in the presence of alkali, that is VP reagent 1, 40% KOH, and atmospheric oxygen. So to provide atmospheric oxygen, we'll be vigorously shaking the tube after addition of the VP reagent 1. You have to vigorously shake the tube. So in the presence of alkali and atmospheric oxygen, acetone will be oxidized to diacetyl. This diacetyl will react with the VP reagent 2, alpha naphthol, to give the red color. So positive reaction is shown by Klebsiella pneumoniae, negative shown by E. coli. Next one is a nitrate reduction test. So this test, it will detect the presence of nitrate reductase enzyme present in the bacteria that reduce nitrate to nitrite or free nitrogen gas. So the media that we use is nitrate broth, and we will inoculate the organism onto the nitrate broth, which contains nitrate, potassium nitrate. And if the organism contains nitrate reductase enzyme, potassium nitrate is converted to nitrite or nitrogen gas. And next day morning, after overnight incubation, we are adding the nitrate reagent. It is sulfanilic acid and alpha naphthylamine. So we are adding this reagent. If it is positive, the medium will be changing to this red color. This red color is a positive reaction. This is the negative reaction. So example, all the members of family Enterobacteriaceae nitrate test positive. That is, it will reduce nitrate to nitrite. Then the next one is phenyl pyruvic acid test. This test is the prototype test that we do for the tribe protein. This test is done for members of the tribe protein. So we are testing the presence of the enzyme phenylalanine deaminase. If this phenylalanine deaminase enzyme is present, it will deaminate phenylalanine to phenyl pyruvic acid. So if the enzyme phenylalanine deaminase is there, it will deaminate phenylalanine to phenyl pyruvic acid. And phenyl pyruvic acid, when it combines with 10% ferric chloride, it will produce a green color. So what we are doing is, this is the media. So we are inoculating the organism on the media, and next day morning, we are adding 10% ferric chloride solution onto the surface of the media. It is freshly prepared 10% ferric chloride. So if the organism is having phenylalanine deaminase enzyme, the phenylalanine present in the media will be deaminated to phenyl pyruvic acid. So this phenyl pyruvic acid, it will react with the 10% ferric chloride to produce a green color in the slant. This is how the positive reaction will look like. So these are the conventional biochemical tests. These are the major conventional biochemical tests that we do. Now we move on to the automated systems for bacterial identification. So these automated systems, they have an advantage that they are faster. We can get a result faster. So for conventional methods, we need to perform the biochemical test, and next day morning only, again, we are reading the reaction. So we are getting a colony of the culture, the bacteria on the culture media, and we are doing the biochemical test. Again, one more day is needed for identifying the bacteria. But in automated systems, the result is faster. Once you get a colony, you can take the colony and you can keep it in the, you can make a suspension, or based on the manufacturing instructions, we can prepare the suspension and load it in the machine, and the machine will give the result within that day itself. So that is how automated systems work. So it gives a faster result, and it will identify wide range of organism, and the accuracy is also high. So one more thing is, in conventional biochemical test, we can't do the biochemical test always for certain organisms such as anaerobes, fastidious organisms which are difficult to grow. So in such cases also, the automated test can be used. We can use automated tests for these organisms, difficult to grow organisms. The examples of the automated test are MALDI-TOF, that is matrix assisted laser desorption ionization time of flight mass spectrometry. Then Vitek 2, then Phoenix. All these are automated identification and antimicrobial susceptibility testing methods, automated test. Then microscan walkaway system. This is also another automated identification and antimicrobial susceptibility testing system. These are the names of the manufacturers, manufacturing company. Now, Malditov, it can identify bacteria, fungi, and mycobacteria with a turnaround time of few minutes and with absolute accuracy. And it is a mass spectrometry that is done. The expansion is matrix assisted laser desorption ionization, time of flight mass spectrometry. So two systems are commercially available, which are named as Vitec MS and Biotyper system. These two systems, it works on the principle of this matrix assisted laser desorption ionization, time of flight mass spectrometry. So the principle is it will examine the pattern of ribosomal proteins present in the organism. So it will identify the pattern of the ribosomal protein present in the organism. So first you have to prepare the sample. So the colony of the organism is taken and it is taken on a slide, on the well of a slide. So for performing Malditov, there will be a slide available. It has got certain wells. It has got wells on it. So the colony is smeared on these wells and 1 drop of matrix solution is added on it and it is mixed well and loaded onto the machine. After loading, what will happen in the machine is it consists of 3 chambers. One is the ionization chamber, second one is the analyzer chamber, and the third one is the detector chamber. So in the ionization chamber, based on the mass of the so ionization will happen, and based on the mass of the substance, it will be moving on to a analyzer tube, and there it will be analyzed and it will be detected by a detector machine. So the ribosomal proteins of various organisms are preloaded onto the system. So the system will be comparing the ribosomal protein pattern of the organism under analysis and it will give the result. This is the pictorial diagram. So on colony is smeared onto the well of the slide. This will be the slide and it has got wells. So we are smearing the colony onto the slide and 1 drop of matrix solution we are adding. Then slide is loaded onto the system. This is the machine and there is an ionization chamber. So laser, based on the laser, it is laser assisted. So there is a matrix which is created. We are adding the matrix solution. So a matrix is created and the ions are accelerated in the electric field. It will move on to the flight tube. This is the analyzer tube. And here, based on the mass, it will be separated. That is why time of flight, the time it takes to move through the tube. This is a flight tube. So based on the mass, it will move, and finally in the detector, the signal processing. Then the Vitec 2 automated system, it will also perform both identification and antimicrobial susceptibility testing. It uses certain colorimetric reagent cards. So Vitec 2 and Vitec MS is different. So Vitec 2, in that it will use certain colorimetric reagent cards containing 64 wells. There will be a 64 well card. That card, the single card is used for one organism identification. So the 64 wells means different biochemical tests are there in the each wells. So individual test substrate is there. So we are making a suspension of the organism and so this will be the card. We are making a suspension of the organism and through a tube which is attached will be by the capillary action, the suspension of the organism will be moving on to the card. It will move through the wells and each well will be having certain test substrate and biochemical reactions will happen. So the pattern of the colors which is coming from the card, that is detected. And that is how, based on the pattern, you will identify, the machine will give the result. And the result of identification, it is usually available within 4 to 6 hours. Then there are certain automated blood culture techniques available. Example is BacT Alert 3D. BACTEC by BD Diagnostics. All these are examples of blood culture, automated blood culture systems. So there is continuous monitoring of the blood culture tubes. In conventional blood culture technique, there is no continuous monitoring. We will be doing the subcultures alternate day. Usually alternate days we will be doing the blood culture subculturing on the solid media. In these automated blood culture techniques, since there is continuous monitoring, whenever there is a positivity signal that is generated by the machine, we can take out the bottle from the machine, we can do the gram staining, we can identify the organism. So it will periodically monitor the blood culture tubes for microbial growth every 10 minutes. And the composition of the broth is usually tryptic soy broth or brain heart infusion broth, and polymeric resin beads will be there. It is to neutralize the antimicrobials which are present in the blood specimen. And the specimens that can be used in these blood culture bottles are blood, bone marrow, and even you can use sterile body fluids also in these blood culture bottles. It is more sensitive, that is, the yield is higher and less time is required. It is less labor intensive. The examples are bacterial 3D. So this will be the appearance of the machine. This is the bottle. Inside the bottle, through the lid, you are inoculating the blood, and you can keep load inside the machine. And once there is a sufficient growth of the organism inside, based on certain principles such as the carbon dioxide production and all, the machine will be beeping. The machine will give a beep sound. You can take out the machine and you can take out the bottle and do the subculture and identify the organism. So when bacteria multiply, it will produce carbon dioxide. It will increase the pH. So there will be a change in color of the blue-green sensor at the bottom of the bottle to yellow. You can see here. So this is the positive. So normally the bottle bottom will be having a green colored base. So it will be changing to yellow color when there is a positive, that is growth is there, because carbon dioxide is produced. It will change the pH and it will change the color of this base. And again, because of this color change, the machine will be beeping. So that is colorimetric detection of the growth. Another one is bacterial birth show. Almost the same principle. Then BACTEC. BACTEC means it is fluorometric detection. That is, oxygen sensitive fluorescent dye is present in the medium. So when there is less amount of oxygen, the fluorescence will be affected. So it is fluorometric detection of the growth. Then the disadvantage of automated system is, obviously, it is a high cost of the instrument and inability to observe the colony morphology, because we are not using the solid medium. We are using liquid medium, and we are not able to initially identify which organism it is. Once you subculture only, you are able to identify the organism. So that finishes identification of bacteria. Thank you.